All right, good morning, everyone. So on last class, we started discussing cell division and mitosis and meiosis, and we went through some of the facts or to know about the cell division and why it is absolutely necessary. We talked about the idea or the fact from the cell theory that all new cells are created from pre-existing cells. And so we took a walk through the eukaryotic cell cycle to look at the process of mitosis, right? And we defined mitosis as a series of steps in which the parent cell transmits genetic information to daughter cells, right? And so this is a process that occurs in order to create body cells. All right. So we're clear that these new cells that we're making has the responsibility of accurately duplicating the genetic material that exists in the parent cell. Right. And that is important when we start thinking about the transfer of genetic information over to the daughter cells. We want to make sure that all of these genes, right, that are found on our chromosomes are accurately duplicated so that these newly created daughter cells are an exact replica of the parent cell. All right. And so we talked about the chromosomes and the fact that humans have 46 chromosomes in our somatic cells. And so we walked through the eukaryotic cell cycle. Right. Just a real quick recap for the cell cycle. There are two main phases. We talked about interphase, and then we talked about M phase, right? Collectively speaking, we said that interphase comprised of this long sort of preparatory period for the cell in preparation for division. All right, so interphase is characterized by gap 1, G1, S, and G2 phase. All right. So this is the cell growing, obtaining nutrients, metabolic potential, synthesizing or duplicating the DNA, and being prepared for nuclear division. M phase is the phase in which the cell is actually going to divide. This takes place during cytokinesis. Okay, during cytokinesis, the cytoplasm of that parent cell, right, that has undergone these stepwise modifications and preparation, during cytokinesis, the cytoplasm actually splits off into two new cells. All right, and so that's kind of where we left off on last class. We walked through those phases of the cell cycle, and then we looked at M phase, mitosis. All right, we said the mitosis is characterized by PMAT, right? Prophase, metaphase, anaphase, telophase, and then cytokinesis. And so today we want to look at another type of cellular division that's a little bit different than mitosis. It has a different purpose and a different outcome. All right. So real quick here with mitosis, we ended up establishing the fact that mitosis produces two daughter cells, right, that are genetically identical. That's what you should have in your notes regarding mitosis. Okay, it is a cell division process that results in the production of daughter cells that are identical to the parent cell. Okay, so the purpose of mitosis is to produce identical copies of the parent cell. All right, these are our body cells that are being produced. All right, your next cellular division is, we went through these already. We went through the events of mitosis and we established the results are two cells that are genetically identical to the parent cell. Okay, so what that means is that each daughter cell receives exactly the same number and kinds of chromosomes that the parent cell has. Okay, today we're going to look at another type of cell division that is necessary for sexual reproduction. Okay, our objectives today are as follows. We should be able to differentiate between asexual and sexual reproduction. We should be able to distinguish between haploid and diploid cells. We should be able to define homologous chromosomes. We should be able to explain the significance of meiosis and describe the processes. And then we should be able to then contrast mitosis and meiosis, emphasizing the different outcomes. All right. So we said the purpose of mitosis on last class was to produce new body cells. Okay, meiosis, the sole purpose of meiosis in cell division is to produce sex cells, gametes. Write that down. These sex cells are necessary for sexual reproduction, one of our characteristics of life. So, reproduction, we're clear that we absolutely must continue reproducing in order to continue to perpetuate a species. There are two basic types of reproduction in eukaryotes. Jot these down: asexual reproduction and sexual reproduction. Asexual reproduction requires a single parent. Okay, what's happening here is that a single parent splits buds or sort of fragments into two, creating new individuals. All right, this is mostly due to a type of mitotic division. All right, new individuals are going to inherit the same genes and traits that are identical to that of the parent. All right, this is a process that occurs very quickly, very efficiently, as this organism doesn't require a mate. So this is a process that can happen pretty easily and efficiently. Asexual reproduction. Okay. On the other hand, sexual reproduction requires the union of two cells, right, that are called gametes. All right. The scientific term for our sex cells are gametes. So sexual reproduction are going to require two cells in order to take place. Two different parents are going to contribute these gametes, right? Gametes will then unite to form a single fertilized egg known as a zygote. Jot that down, right? So two parents. In our case, as humans, we have mom and dad. Both have different sex cells. We have sperm and egg, right? These sperm and egg will unite to form a single fertilized cell that we'll call a zygote. This process is absolutely essential for sexual reproduction. It requires two parents. It is one of the most distinguishing features between asexual and sexual reproduction, okay? Requires two parents, and the outcomes are going to be significantly different. All right. So the results of reproduction, asexual reproduction, jot this down, is basically going to produce genetic clones. Okay. You're simply going to create an exact replica of the cell you started with. Okay. In sexual reproduction, though, this process results in genetic variation in the offsprings that are created. Right. Each offspring is going to be genetically different. Physically, the phenotype is going to be different as well as its genotype. Excuse me. So we're saying here that the offsprings that are created from the joining of that sperm and egg are not going to be genetically identical to the parent. And that makes sense, right? Because the offspring that is created is essentially going to be a hybrid of both parents. So you're not going to have all chromosomes from mom or all chromosomes from dad. You're going to get 50% from either parent. Okay. So sexual reproduction here, the point we're driving home is that sexual reproduction results in genetic variation in those offsprings. Okay. Each offspring that is generated is going to be genetically different. All right. Just if you think about, if you have siblings, right? If you have 2 to 3 siblings, you guys are all going to be of the same parents, right? But you're all going to have a different combination of chromosomes from both mom and dad. So the features that you exhibit are going to differ. I mean, there will be certain features that people will say, oh, you look just like your sister, you look just like your brother, you look just like mom or dad. Well, that's true because that's where you're getting that genetic information from. But in terms of your siblings, you are going to exhibit various characteristics that are going to be a little bit different because you all have different combinations of genes. Okay, so real quick, for those of us that are visual, sexual reproduction over here on the left, what's happening? We need 2 parents. Okay, we need mom and we need dad. All right, the result, the offspring that's created during sexual reproduction is going to be a mixture or a hybrid of both parents. Okay. This is sexual reproduction. We need two parents. The genetic information from two parents are going to be shared. This offspring is going to be a hybrid. Asexual reproduction only requires a single parent. The offspring that is created is going to be identical to the parent. What question do we have about that? Any question?
No, ma'am.
Awesome. So real quick, for a point of comparison, there are some advantages and disadvantages to both of these types of cellular division, right? We're saying here, jot this information down. This is some basic information about sexual and asexual reproduction, or snap a picture of this table. Sexual reproduction, we're clear that it requires two parents. And so what's happening here is that the sperm and egg is fertilizing one another to form our zygote. The offsprings that are created are going to be genetically unique from the parent, okay? But it's going to contain a random combination of genes. So you're going to get a random distribution of genes from both mom and dad. Sexual reproduction is used by humans and other types of plants and animals. Some of the pros, again, is that we're producing genetically different individuals, right? Increasing the survival rate in the population. So genetic variation is a good thing. Okay, genetic variation provides various survival advantages over time. When we start thinking about evolution and being able to adapt, right? Over time, the genetic variations that we inherit from generation to generation is absolutely critical to our survival. And so they are a good thing. One of the downsides for sexual reproduction is that it's a very time-consuming process. All right. It requires a lot of time. It requires a lot of effort in order for this process to occur. And it can be thought of as a negative, right? It takes a long time. All right. Asexual reproduction, on the other hand, we're saying that it requires only one parent, right? One organism. A single organism then makes a copy of itself, essentially, and then it divides. All right. So we're talking about a process very similar to cloning. Duplicate the genetic material and then split off into two. The offsprings that are created from asexual reproduction are genetically identical to the parent. This process is used by fungi, yeast, bacteria. Okay, so binary fission is an example of asexual reproduction. Budding, used by yeast, is an example of asexual reproduction. Pros. This is a very efficient process. It happens very quickly. It happens very rapidly. Okay, it doesn't require a whole lot of effort. One of the downsides, though, is that if the parent organism contains any genetic defects or abnormalities or deficiencies, all of its offsprings are also going to inherit those same genetic defects and deficiencies. And so that's definitely not ideal. Okay. And so again, when we're thinking about these processes, there are pros and cons to each. All right. Some happen fast, some take a little bit longer than others. But the fact that asexual reproduction basically results in a cloned individual, any abnormalities or deficiencies that are in that parent organism are going to be passed down to that daughter cell. So that's certainly a negative. All right. So when sexual reproduction takes place, there's an exchange of chromosomes, right? And so each chromosome that we have exists in pairs, if you will, right? So we call these homologous chromosomes, okay? Each chromosome in our somatic cells has a partner chromosome, right? So two partner chromosomes known as homologous chromosomes are going to be similar in size, shape, and position of that centromere, right? And so these homologous chromosomes are going to carry the same genetic traits, right? Remember, we looked last class at the layout of the human karyotype, and we said that the human chromosomes are identified by number, and we know which genes are found on which chromosomes and which traits they encode. And so these pairs of homologous chromosomes are going to contain the same genetic information on each. All right. The information may not be identical, but they're going to carry the information for the same traits. All right. These traits, for example, are eye color or hair color. You know, there can be variations in your eye color. All right. So your homologous chromosomes may or may not be identical. So the homologous chromosomes, we're saying, contain DNA that codes for the same genes. Okay. We talked about chromosome number in our somatic cells. We said that we have 23 pairs of chromosomes. All right. So if a cell has 2 sets of chromosomes, that cell type is said to be having a diploid number. Write that term down, diploid. Okay. Chromosomes have 2, if the cell has 2 sets of chromosomes, that is a diploid chromosome number. Okay. For our somatic cells in humans, the diploid chromosome number is 46. 2 sets, 23 pair. Okay. That's what we're going to find in our somatic cells, a diploid chromosome number. However, if the cell only has 1 set of chromosomes, okay, it is said to have a haploid, think half, haploid. The haploid chromosome number for humans is 23. So which cell types in humans have a haploid number? We have 2 general types of cells in our bodies. We have somatic cells and then we have sex cells. Somatic cells are our body cells. They are responsible for our overall existing growth, development, all of those different characteristics. And then we have our sex cells, which are responsible solely for reproduction. If our sex cells, if our somatic cells have a diploid chromosome number, which cell types in our bodies have a haploid number?
Sex cells.
Sex cells, thank you. All right. And so real quick, this is just a schematic. If we're looking at the chromosomal makeup of our cell, diploid cells have a 2 copies, okay? 2 sets of chromosomes. Haploid has half. All right. So reproduction, right? So what's happening during reproduction is basically we're getting the union of the sperm and the egg. All right. Each gamete has a haploid number of chromosomes. Okay. This means that each cell contributes 1 set of chromosomes. During this formation or this union, we get what we call a restoration of the diploid number because this fertilized egg, this zygote, now has 46 chromosomes. Okay. The process of mitosis then will happen where we create 2 new cells with 46 chromosomes. Okay. That's what happens during mitosis. All right. Again, this is just another illustration for those of us that are visual. Okay. Diploid cells are going to have 2 copies of each chromosome. So any chromosome that's present in a diploid cell will exist in pairs. Okay. In diploid. Okay. Haploid cells are going to have half that number of chromosomes. So meiosis, what's happening during meiosis? Meiosis literally means to make smaller. Okay. Meiosis is then a cell division process that reduces the number of chromosomes by 1/2. Right. So we're looking at a process where this diploid cell is undergoing 2 rounds of cellular division in order to yield 4 haploid cells. Okay. This is in complete contrast to mitosis. In mitosis, we saw 1 round of cell division. We created 2 daughter cells, and the 2 daughter cells were diploid. Write this down. With meiosis, we are undergoing 2 rounds of cell division. We're going to create 4 daughter cells. Write that down. And these daughter cells are going to be haploid in nature. That's what you need to know about meiosis. This is a reductive cell division. Okay. What questions do we have? All right, so meiosis, what's happening? This is the process of sexual reproduction. There's a phenomenon happening known as crossing over. Okay, these chromosomes are separating and dispersing randomly to haploid daughter cells. Okay, this process happens randomly, right? We have no control over the disbursements of the chromosomes and traits that are being passed down to these daughter cells, right? And we said that meiosis occurs over 2 steps. Okay, so we have meiosis I and we have meiosis II. Okay, meiosis I, 2 haploid cells are produced from a diploid cell. Okay, during meiosis II, those same 2 haploid cells are going to be further reduced, and these chromosomes separate into chromatids. All right, I have a visual. But real quick, let's make sure that we understand some important differences in the events of meiosis. The first difference that you need to be familiar with is that meiosis involves 2 successive cell divisions. They're going to produce up to 4 daughter cells. Now we need to make sure that we're clear that although we're undergoing 2 rounds of cell division, we're only going to duplicate our genetic material once. Okay. Each of the 4 cells that are produced during meiosis contains a haploid chromosome number, right? Because we're clear, the cells that we're producing during meiosis, these are gametes. So it makes absolute sense that these gametes only contain a haploid number of chromosomes. Because why? What are gametes used for? What are gametes used for biologically?
Reproduction.
Sexual reproduction, right? So each of these new cells that are produced during meiosis are gametes, which means they are going to have a haploid chromosome number. Each haploid cell that is produced are going to have a unique combination of genes that they've just gotten from mom and dad. These are 4 very important differences when we're comparing mitosis and meiosis. Meiosis involves 2 successive cell divisions. We're going to produce 4 daughter cells. These 4 daughter cells are going to be haploid in nature, and they are all going to be genetically different. Okay, let's keep going. So for those of us that are visual, I've got a side-by-side of what's happening during these cell division processes. We've got mitosis over here on the left. We've got meiosis over here on the right. Okay, during mitosis, right, we start with a diploid cell. Okay, these somatic cells have a diploid chromosome number, 2 copies of each. Okay, at the start of mitosis, we're going to have 1 parent cell with a diploid number. At the end of mitosis, you're going to end up with 2 cells that are also diploid. So we've basically cloned these cells over here for mitosis. Meiosis, the process is a little bit different, okay? We start with a diploid cell, but now we're trying to make gametes, right? So we have to undergo 2 rounds of cell division, 1 and 2, okay? These 2 rounds of cell division are going to look just like mitosis, right? But our end goals are going to be different, okay? So we got meiosis I, where we end up with our 2 diploid cells created. And then we have meiosis II, where we end up with our 4 haploid cells being created. Jot this schematic down. If you see these schematics, you should be able to easily identify which processes are taking place here. Mitosis has one round of cell division where we end up with two genetically identical daughter cells. Meiosis requires 2 rounds of cell division, okay, 1 round and 2 rounds, and then we end up with 4 genetically different haploid daughter cells. All right. So what's the purpose of meiosis? In your notes, you should now have that meiosis is required in order to produce sperm and eggs that are needed for sexual reproduction. Okay, so the sole purpose of this process of meiosis is to produce gametes. Okay, the goal here is to produce daughter cells with half as many chromosomes as the starting cell. So no matter what, when meiosis is complete, we should have a daughter cell that has a haploid number of chromosomes. Okay, these phases of mitosis, they look just, meiosis, it looks just like what's happening in mitosis. Okay, so meiosis is a 2-step division process, as I said earlier, right? Meiosis I, we've got the separation of those homologous chromosomes. Meiosis II, we've got these sister chromatids now separating off into these haploid cells. Okay, so meiosis I, meiosis II, they look just like mitosis. Okay, so again, prior to meiosis happening, the cell is also going through interphase, right? So we looked at the cell cycle already. We know that those preparation periods don't change. Interphase G1, S, and G2, right? And then meiosis I begins. Meiosis I looks just like mitosis. Meiosis I, the events of meiosis I are the same as that of mitosis, okay? PMAT. Prophase, metaphase, anaphase, telophase. Okay. Prophase, we see the chromosomes start to condense and pair up and they're becoming visible. Metaphase, these homologous pairs of chromosomes are going to align up toward the midplate or the center of the cell. Anaphase, these paired chromosomes are going to separate and move towards opposite poles. Okay. Each group that's moving in opposite directions are going to receive a sort of a random distribution of chromosomes. Okay. Telophase, we have nuclear envelopes starting to form around each of these sets of chromosomes out at opposite ends of the pole, and then cytokinesis occurs, right? And so this looks like exactly what's happening during mitosis. All right, so that's the first step of meiosis, meiosis I. Right? Then meiosis II takes place. So this is another round of division, right? Where we're seeing these chromosomes condense and line up, PMAX happening again, right? But now we're going to end up with these chromosomes are going to be split again, and cytokinesis will occur again, and we will have 4 haploid daughter cells at the end of meiosis II. Okay. So why do we care? We care because meiosis generates genetic diversity. Genetic diversity provides survival advantages. Okay, so the exchange of genetic material between homologous chromosomes in meiosis I, right? We've got this random alignment of maternal and paternal chromosomes. There's no rhyme or rhythm to the distribution of chromosomes into these daughter cells. Okay, it's all due to random alignment. I'm going to stop here. I know we're almost out of time. Jot these questions down for homework. You're going to complete them in your notebook. These are some important concept review questions here. You should now be able to explain the significance of mitosis. Explain the significance of meiosis. Then identify 3 to 4 distinct differences between these two processes. And then lastly, I want you to describe what's happening during crossing over. Okay, take your time, use your textbook, use your the World Wide Web, whatever resources you need to further synthesize this information. All right, we're going to stop here today. You have a few minutes remaining in class that you can go ahead and get started on these questions, and we will continue on next class. What questions do you all have for me?